It is my pleasure to introduce the speakers for this session. So we have four great speakers, Jill McLellan from the University of Wisconsin, Brad Fortune from Devers Eye Institute, Jason Meyer from Indiana University, and Tasneem Sharma from Indiana University. The learning objectives of this session is to identify large animal models of glaucoma and understand their strengths and weaknesses, understand best imaging practices and data interpretation, recognize and understand best practices and potential for using stem cells in glaucoma research, as well as identify ex vivo model systems to study glaucoma, including strengths and weaknesses. So our first speaker, I'll turn it over to Gillian.
Yes, Dan, Diane, I'm here. I got this. So, good morning, everyone. So just a little bit of background. I'm actually a veterinary ophthalmologist, and I work in the vet school at University of Wisconsin-Madison, but my lab is in the medical school. Just quick disclosures, I am a consultant for eye care who make the TonoVet and TonoLab tonometer, and also for an electrophysiology company and a contract research organization. I'd just like to, before I get started, thank everybody in my lab and all the funding agencies that have supported my work. Large animal model work is very, very, very much a team science and very, very costly. So I'm very grateful for all that support. And just a quick call out to Anjali and Virginia that are here, and we'll have a poster. Each have got a poster on Thursday, so if you want to ask questions about animal models of glaucoma, you can stop by and talk to them because they do the stuff. In terms of background, I'm largely going to focus on what I call large animal models of glaucoma might be different from others. As a veterinarian, large animals are actually things like cattle and sheep and pigs and llamas and camels. So dogs and cats and rabbits we consider to be small animal models. So there is a little bit of confusion and disconnect with the veterinary community there. But really I'm going to focus on rabbit models and particularly feline and canine genetic models of glaucoma that myself and another veterinary ophthalmologist, Andras Komaromi from Michigan State University, maintain colonies of. So the big advantages of these particular non-rodent models of glaucoma are that rabbits, cats and dogs are domesticated species. They're very easy to handle. They're amenable to non-invasive imaging that you're going to hear about from Brad in a few minutes. And they have a globe size compared to rodents that's much more comparable to that of humans. So a lot of the diagnostic tools that you'll hear about, so things like OCT, for example, they work perfectly well in things like dogs and cats and to some extent as well rabbits. Now the caveat, there's loads of other species that I'm not going to cover today, and Brad will be talking a little bit more about non-human primate models of glaucoma. So in terms of the background, what is it that we're interested in when we're looking at glaucoma models? Clearly there's two camps typically. There's going to be those that are interested in the role of the optic nerve head, the retina, and also the central nervous system in the brain. They're going to be interested in the cell population in the optic nerve head and the retina, and also the biomechanics of the eye. Biomechanics of the optic nerve head is going to be really important when we're studying glaucoma models. We are interested, if we're interested in the front of the eye, in the role that intraocular pressure plays in the development of that subsequent neurodegenerative disease. So we're going to be interested in the structure and function of the aqueous humor outflow pathways. And so some of these large animal models are going to be useful for that as well. Eye size, as I said, is going to be important. So it's going to be important because that impacts things like biomechanics. And it also is going to impact pharmacologic studies. So if you want to do studies of the effects of a drug on aqueous humor dynamics and intraocular pressure, or a drug that you want to get to the back of the eye to be neuroprotective, then the size of that globe of your model is going to be really important. So as you can see from that little picture here along the bottom, we can see that there is a real spectrum from this tiny little rodent model with their teeny tiny little eye that we can see over on the right-hand side, and they tend to be short-lived species, so there's a spectrum of longevity from the non-human primates through cats that can live to be 15, 16, up to 20 years of age, dogs where the typical lifespan is going to be about 12, and then pigs, which if they weren't in a production system could live to be quite old as well, then rabbits and then rodents. And there's also a spectrum in terms of the response to inflammatory insults to the eye. So it's, if you think about this from an evolutionary perspective, if you're sort of a herbivorous species, for example, and you poke yourself in the eye with a piece of straw, it's going to be more important that you just seal up your eyeball and can continue to go about your day eating for a living compared to an animal that might have to capture prey or function with tools as a primate would. So there tends to be a great dampening of that inflammatory pathway in some of these higher order mammals that we consider. And then there's going to be some quite significant structural differences in the optic nerve head that we can see in these digests on the slide as well. So just quickly, the feline model that is going to be most represented in the literature is the one that we have in our lab. It's the only spontaneous model of feline glaucoma that is maintained in a colony setting, and that's maintained at UW-Madison. Now, these animals have a mutation in a gene called LTBP2, which is a matricellular protein, so they have abnormalities in their ocular extracellular matrix. And Odalis Tournay, that you can see, that's a former graduate student from my lab, did a lot of studies looking at the trabecular meshwork, and we see very early changes in trabecular meshwork biology within the first few weeks of life in these animals that subsequently causes a progressive increase in intraocular pressure. And that in turn is associated with a progressive loss of retinal ganglion cell somas and axons in the optic nerve. But one thing to be aware of that we can see in this graph over here is that there's a lot of variability when IOP goes up. There's a great deal of heterogeneity, and that's going to be a common theme that you're going to see in these non-experimentally induced animal models of glaucoma. So the mutation, as I say, is in a gene called latent transforming growth factor beta binding protein 2, or LTBP2. And this mutation really introduces a stop codon. They actually don't form any functional protein. These animals, as I say, end up with a very early life glaucoma that's actually the ortholog of a human form of glaucoma related to LTBP2 mutation in people as well. So some of the strengths of this model is that it's early onset. So I don't need to keep animals in the lab for 2 years before they develop an increase in intraocular pressure, which of course would be very costly. They have an optic nerve head structure that very closely resembles that of humans. And Kazuo Oikawa, a postdoc in my lab who did his PhD in my lab, has done a great deal of work characterizing the feline optic nerve head and looking at its response to insults in glaucoma. So not only do they have a extracellular matrix structure that's very similar to that of humans, but they have differences in cell populations that really mimics what we see in the human and non-human primate eye. So their cell and molecular biology really is very highly context dependent, even within the optic nerve head itself. And so that's going to be an important thing when we look at other species that don't have that same level of organization of their optic nerve head. Some of the weaknesses, however, we touched on that heterogeneity of the IOP profile and their phenotype, but I like to see that and as something that's potentially an opportunity. So what are some of the other potential genetic drivers, for example, that might be resulting even in that very homogeneous population, this heterogeneous profile that we're seeing? We do sometimes see a displacement of the lens in these eyes, and that's something that can be a big feature of some human families with this mutation, or with a mutation in this gene. And there's a lot of resources and housing requirements that need to be met to be able to ethically keep a large animal model like this. And the same is going to apply to dogs. And there's going to be ethical considerations because they're damn cute and they're domesticated animals, and we all love them. So dealing with an animal model like that brings all kinds of ethical concerns that we can't take for granted. Their outflow pathway is a little bit different. The models that we're going to talk about today, dogs, cats, and rabbits, don't have a Schlemm's canal, but they do have a vacuolating endothelium in an angular aqueous plexus that takes the place of Schlemm's canal, and they have a pretty robust trabecular meshwork for study, however. In the canine model, again, it's an extracellular related, matrix-related protein called ADAMTS10, which is involved in matrix metalloprotease activity. And that has been recognized in beagle dogs, and Andras Komarov maintains a colony of those dogs. Previously, they were at Florida with a professor called Kirk Gillat, and now they're maintained in Michigan State. So again, it's a colony that is available for study. They get a gradual, slow increase in IOP that occurs, you can see there, over about the first 60 months of life. So it's very gradually progressive over the course of their life. And they maintain an open iridocorneal angle, and they develop optic nerve head cupping. And since the gene was first identified in beagles by Rachel and John Kuchty back in 2011, a number of other breeds have been found to have either ADAMTS10 or ADAMTS17 mutations. So these are quite common causes of open-angle glaucoma and also lens luxation in dogs in the general veterinary population that I see in my clinic. But we also see mutations in a similar gene, ADAMTS17, which are even more common. And we find, interestingly, that in these breeds of dogs that size matters, so there's probably some genetic modifiers that are also associated with their diminutive stature. So it tends to be little short-legged breeds that have these mutations. So some of the strengths are that, we don't need experimental interventions. They're amenable to handling. They have a lamina compressiva structure similar to humans. And they're really an established model. Beagles are used very widely in pharmacology and for translational research for bringing drugs to market. But there is also a spontaneous disease called angle closure glaucoma in the general veterinary population that's much more highly prevalent than open angle glaucoma in dogs, and it's an underexploited gold mine. And I personally am looking at the genetics of that form of glaucoma. In terms of weaknesses, there's a variable time of onset, there's heterogeneity, just as we saw in the feline model. They have extensive myelination of their prelaminar optic nerve, so it's actually quite difficult to image lamina creprosa per se. And again, they are quite expensive to care for and to maintain in a colony setting. And again, those ethical considerations, these cute little dogs looking up at you that you are having to maintain in a colony. And now, canine glaucoma is not the same as human glaucoma, however, because there tends to be an ischemic insult that's also associated with glaucoma in dogs, even at relatively low elevation in IOP, not just because of acute angle closure, but even an open angle glaucoma. And that's probably one of the things that we learn from looking at the comparative aspects of dog optic nerve head, is that in other species like cats, like in people and non-human primates, the short posterior ciliary arteries that feed into the retina are actually normally protected within a flange of scleral collagen surrounding the optic nerve head. And in dogs, there's not that protection. Those blood vessels are actually within the canal. And so when you've got any elevation in intraocular pressure, you're collapsing those blood vessels. So we end up with retinal ischemia in these dogs. So it's a little bit different. Talking about weird optic nerve heads, we'll move on to rabbits. Rabbits are really advantageous because they take up a little bit less space. They are widely used in farm tox studies. They're easy to breed, like rabbits, and house and handle. And you can have pigmented and non-pigmented albino strains, which can be important when we're studying drugs. There have been a couple of experimental studies that have shown either using viscous substances or microbeads injected in the eye, you can actually induce an elevation of intraocular pressure, and a lot of the caveats that you've heard about in microbead models will be relevant to rabbits, with the other disadvantage that they are quite an inflammatory model. They tend to get inflamed, they tend to scar a lot when you insult the eye. But they also can have spontaneous elevation in IOP, and just recently someone finally took these spontaneous cases of rabbits and New Zealand whites that had high IOP, and they just kept breeding. They found enough of them that they could breed those rabbits, and they have actually finally established a colony of New Zealand white rabbits in Japan that have a genetic basis for where all of the animals are affected with glaucoma. Now, one of the disadvantages, again, as you can see with this IOP, it's really widely variable. So because it's a spontaneous, likely genetic model, we're seeing this high variability in intraocular pressure, which makes it difficult to show effects without large sizes of sample sizes. They have a narrow angle normally, and they have a really weird optic nerve head with no real true lamina cribrosa. So that might be an important thing to bear in mind. If you're trying to study the lamina cribrosa and the optic nerve head in glaucoma, perhaps a rabbit wouldn't be the best model to use. In terms of pitfalls to avoid with any of these animal models, you really have to understand the differences in anatomy and physiology. Now you know who to ask if you're considering using one of these models, because that's something I've been studying for over 3 decades. Often there's a lack of rigor in terms of validation of research tools to use for these animals. So you need to validate things like a tonometer or electrophysiology or anything that you're using in a novel model. And you really need to make sure you're monitoring these animals very closely for potential adverse outcomes. It's a bigger deal if a cat has a painful eye than nobody paying any attention to a poor little mouse. Studies will ultimately often be underpowered just because they're so expensive to carry out. So one way to consider that you can empower your studies by using longitudinal studies, which I'll be handing over to Brad to talk a little bit more about that. So in terms of the take-home message, what models are the best to use? It really depends on what you're studying. Don't forget the 4 R's: animal use, replacement, reduction, refinement, and I've added one R to those 3 R's for animal use, which is rigor. You've really got to do a good job when you're using these large animal models. Perhaps the 5th is raise the bar. So with that, I'm going to hand over to Brad, and he's going to talk a bit more about some of these longitudinal studies, and then we'll have some unanswered questions at the end, hopefully.
All right. Thank you, Jill.
Thank you so much, Jill, and to organizers for the invitation to present to you today. So I was handed this title, Best Ways to Image and Select Outcomes, and I decided I didn't really like the word best because, consistent with the theme of the morning, it really depends. It depends on what your goals are, what your scientific goals are, what your hypotheses are, A, and B, because 5 minutes from now, someone somewhere is going to discover something that made the previous best thing obsolete. Well, maybe not obsolete, but not as good. Okay, so I've changed the title to Imaging Selected Outcomes, in part also because we only have 12 minutes. So there are 2 disclosures that are important. I will show you some data from a study that we did on behalf of Perfuse Therapeutics that was presented here at a previous ISO BrightFocus Glaucoma Symposium. And also I'll show you lots and lots of images that we obtained using the Heidelberg Spectralis instrument. So sort of by way of outline and overview of what might come later, more entertaining than words, that is pictures, I'll just start with this slide that organizes thoughts into 2 main categories: imaging structure, anatomy, and imaging function or physiology. And so I'm going to try to walk through in the actual images I show you, a little bit like going from a 1960s Volkswagen Beetle to one of these billionaire rockets, personal ship into space. That is, I'm going to go from color fundus photography all the way to adaptive optics, OCT, and SLO. Anyway, to start briefly, photography is widely available, of course, and can be enhanced by, you can do fluorescence photography with the right excitation and emission filtering, of course, changing the illuminant, that is. And it's amenable to lots of different analyses, especially of the macrovasculature, the large vessels, which have high contrast and color contrast, et cetera. OCT, on the other hand, has really revolutionized the world of imaging the eye and other parts of the body because of its 3-dimensional capabilities. And now a functional extension of OCT is OCT angiography. That's a non-invasive, dye-free method of visualizing the vasculature, including down to the very smallest capillaries. And then, of course, scanning laser ophthalmoscopy, often used in confocal mode, but so many interesting studies around the world using the non-confocal, what we usually block out using confocal pinholes. There's so much light that actually contains image-forming contrast from what we usually block out that it opens up a whole world of really interesting stuff. And there's a video at the end to show you one example of that. In terms of physiology or function, blood flow is one of our strong interests. And there are lots of ways of doing that. I'll show you some data from laser spectral contrast imaging. There's also now the ability to look using OCT at changes in reflectance intensity or phase, distances of time of flight in the retina. So you can look at function of individual neurons or layers of neurons in the retina using OCT. And that will have to do with typically osmotic swelling. We're also very interested in looking at subcellular scattering dynamics using the OCT. And then I guess it's not unreasonable to say that you can image function in a way by doing multifocal electroretinography. So let's move on to the selected outcomes. So of course, for glaucoma, we're mostly interested in retinal ganglion cells. That's the neuron of interest. And you might want to be counting soma, measuring their density, or perhaps measuring their size. We're interested in their intrinsic optical properties. You might be interested in the dendritic arbor and characterizing its change over time and methods for restoring its integrity. You might be interested in looking at the axons. Axonopathy is an important aspect of glaucoma and a target for lots of avenues for neuroprotection. Similarly, you might be interested in the function of ganglion cells, and you can introduce genetically in mice, for example, or via virus or lipid nanoparticle delivery, again, sort of gene manipulations to introduce calcium reporters, for example, or other contrast agents specific to ganglion cells that would be related to their function, such as measuring axon transport integrity. You might and should probably be interested in what's happening with microglia and macroglia, and there are ways to do that as well. And then I did already mention the vasculature. That's another form of function, be it blood flow or blood retinal barrier integrity. We're really interested also in the interaction between the cardiac cycle, so the pulse that's coming into the eye, and the biomechanics of both the vasculature and the tissue around the vasculature, the retinal parenchyma for that matter, or the optic nerve. And there are some interesting phenomena that occur because of that interaction. Okay, so thank goodness Yvonne O presented this morning about the most complex model of all, the human model of glaucoma. And I will show you a couple of clinical cases. So here is an eye that, despite tertiary care center, Devers Eye Institute care from excellent physicians, this person lost a lot of vision over the course of the 9 years of follow-up in this slide. But here's color fundus photography, right? The most accessible and relatively inexpensive thing. You can do this with a cell phone camera. At year one here, when the field was looking pretty normal, there's a retinal nerve fiber layer bundle defect that I bet you can't appreciate. And that's the point of the slide, in fact, that without those black arrows, you'd be hard-pressed to see it. You'd probably do better in a clinical exam because these are actually one half of a stereoscopic pair of photographs. So the stereo will help. There's a notch in the neuroretinal rim that Yvonne pointed out earlier. Well, you can see that grows over time. The notch gets much bigger. You can see it here. The nerve fiber layer defect is now all the way from there to there. But again, the point is it's really hard to see in the color fundus photograph. People use so-called red-free photography frequently because the retinal nerve fiber layer has very specific characteristics, optical characteristics, and I'll just take a minute to talk about that. So the nerve fiber layer, just for those of you new to the field, is the most anterior layer of the retina, pointing up, unless you're an anatomist, and then it's pointing down. But anyway, in cross-section by EM, again from Hogan et al., the textbook on a human anatomy, you can see that these axons in cross-section, you can see their cytoskeleton. You can see the neurofilament and microtubules, as well as larger organelles, mitochondria, et cetera. And then in cross-section, you can see that these form a highly ordered tertiary structure, a three-dimensional array of cytoskeletal elements that confers three, probably more, but three really useful optical properties that speak to the integrity of the cytoskeleton. One is birefringes. That literally translates to two indices of refraction. So a photon traveling in one axis will encounter many more of the cytoskeletal elements than one that's vibrating in the plane of the fibrils. So the higher apparent index will give you a faster phase, just like you would have in ultrasound. And that can be exploited by polarization-sensitive imaging to study the integrity of the guts of an axon before it's gone. So we find this to be really important as well, to try to study what's happening before it's too late. Most clinical decisions are made after something has happened, like tissue thinning or field loss. And Yvonne showed examples of recovering some visual field sensitivity. But generally, once we lose vision from glaucoma, once we lose the ganglion cells in the axons, we're not getting them back. We look to the future for that sort of thing, but for now, we don't. So we're really trying to develop biomarkers that tell us a little bit about what's happening before it's too late. Oops, I think I went too far. Because another important property is high reflectance and high directionally dependent reflectance and high spectral reflectance, which I mentioned a moment ago, that you can do a red-free photograph and increase the contrast, the visibility of the axons in the nerve fiber layer. Okay, so here's another clinical case. At baseline, you can see an OCT B scan. You can see this fantastic three-dimensional structure, or the axial resolution that it affords. And then some years later, you can see how the cup has deepened and how these little green vectors here that represent what we call the minimum rim width have both changed their angle. They've come more in plane with the Brooks membrane, and they've become shorter. So that's rim thinning and elements of cupping, as we call it clinically. Another typical scan pattern is the circumpapillary B scan, because you can assess all but about 1 or 2% of the ganglion cell axons, those that live inside that little scan area. The green line here is a little small. We'll blow this up in a second. So these are the typical scan patterns used in a clinical setting that can be used in any of the large animal models that are under discussion. So here's a blow up of that. Again, you can see the changes from test 1 to test 7, so that's 3.5 years later at our institution. And you can see how the conformation, the nerve head is undergoing deformation, almost pathognomonic for glaucoma. You can see there's thinning of the nerve fiber layer. And now if you take all 24 B scans that make up this radial scan pattern and present them as a 3D picture, you can see again that the rim has undergone thinning, especially at this one location here. This is the Brooks membrane opening points, those little red dots representing sort of the anatomic boundary of the nerve head. Now, if you represent those two ribbons of tissue, this sort of golden color is the circumpapillary B scan, the nerve fiber layer area, if you will, cross-sectional area, and then the rim tissue area, you can see again that the rim tissue has undergone thinning right here, consistent with the nerve fiber layer defect. This is the supratemporal pole of the disc, and so there's an infratemporal arcuate bundle defect. And these are parameters that you can then track over time and analyze statistically, such as just something simple like using ordinary least squares regression to determine a rate of progression. And you might be interested in understanding what factors confer the risk of the highest rate of progression. Some glaucoma patients, as shown in a previous slide, progress despite really successful management in their IOP. And we want to know what it is about that eye that confers that high risk of a really nightmare scenario, that even under successful treatment, the person is still losing vision. You might be interested in a therapy and whether it modulates the rate of progression or not. Okay, so here's that clinical sort of picture, if you will. This happens to be the infrared SLO image painted onto the OCT surface so that you can see the difference in the rim has undergone thinning at this location here as we went in time. So this kind of representation is interesting. Here's a monkey. This is an older set of scans where it's less dense by a factor of 3 than what we do now. And what I'm just showing you here is a time series showing you over the course of a couple of months how glaucoma will develop. And here comes the cupping. And so, you can see that deformation. We're really interested in what it is that sort of it damages axons as this deformation occurs. And so one of the things is IOP. So here's an example of a reflectance OCT painted onto the surface on your left, and then the angiograph that I mentioned, OCT angiography on the right, as we go stepwise elevating IOP, 10, 20, 30, 40, 50, 60, and back to 10. So let's let that video rip. And here it goes, 10, 20, 30, 40, 50, 60. And you can see how there's some vascular changes. There's certainly confirmatory changes. There's changes in the reflectivity of the ganglion cell axons. And those can be used diagnostically, we hope prognostically, to test ideas about pathophysiology, et cetera. To quantify that sort of thing, you can see how the rim volume in the glaucomatous eyes started off low because, well, they had glaucoma. But the control eyes tested twice each, that I should say the fellow eyes. There's Rob. We find, I would never say that they're normal, but they're excellent control eyes, as this data shows, because over time they're behaving exactly the same as they do all along with some small aging changes, et cetera. They don't lose any ganglion cells, they don't lose any axons. But anyway, their behavior is very different than the glaucomatous eyes, and that's shown in this other slide. Here's the vascular volume changing. So both of these sets of slides show you that at this very early stage of experimental glaucoma in a non-human primate, the tissue is hyper-compliant. I better move on. Directional. You can see that depending on where you enter the pupil, you get a prismatic effect and you change the reflectivity of the nerve fiber layer, and you put the scan out of focus in other areas that really hurt your ability to do a good angiograph. Okay, and we're hoping to leverage that ability to do directional OCT to reveal parts of the lamina cribrosa that we otherwise don't see well on an axial scan. The clock says 0, so I'm gonna move on. Is that right? It must be. I am. Okay, I'm gonna move on. I'm gonna move on because what I really wanna show you is the other end of the spectrum. Here's a video of blood flow, and then in the fellow eye, and you can see over here the blood flow is really damaged and reduced in that area of the optic nerve in a non-human primate. And you can manipulate this. I showed you in a previous ICE BrightFocus meeting with a endothelin antagonist that you can increase blood flow in a dose-dependent manner. So you can image the ganglion cells in the dendritic arbor, as Chris Leung's lab showed many years ago. You can image the microglia. These are genetically encoded reporters in mice. And then finally, just to finish off, I want to show you some work that our newest colleague at the Devers Eye Institute has been doing. Kazuhiro Kurokawa started his lab right in the midst of the pandemic, which was interesting. And he built our laboratory system using, in part, help from the BrightFocus, support from the BrightFocus. So it's been critically important. But the beauty here, I think you can readily appreciate, is the ability to see individual ganglion cells in the living eye. This is the commercial imaging by Spectralis here. And the disadvantage is the isoplanatic patch over which adaptive optics is useful is only about 1.5 degrees or so. And so you can really only do scans. You have to montage to build up a whole area like that, or just do scans over time in that one area. But the thing about laboratory experiments is that you can then get the tissues and validate using, for example, RBPMS immunohistochemistry. At ARVO last year, in collaboration with Kazuhiro and his group, we've all demonstrated that you can see the ganglion cells very well, except those that are tucked up in the undulations of the nerve fiber layer. You can see them well in vivo, but we miss the ones that are tucked up inside the nerve fiber layer. And then we also miss the sick ones in glaucoma. So here's a GCC interretinal thickness map of an animal that had a nice plush baseline and then a superior arcuate defect. And then the AO imaging was done in this spot right here. And it's very difficult to appreciate any ganglion cell soma in that patch. But when we went and go ahead and do the, whoops.
We should probably move on.
Okay.
Yeah.
So that'll be that.
Yeah.
You can see that there are plenty of ganglion cell soma still there. And so back and forth we go between imaging. So you might want to use imaging to study the model, but often will use a model to study the imaging. That's the point I wanted to make. Thank you.
Great. Thank you, Brad.
Oh, I don't.
All right, and our next speaker is Jason Meyer.
Okay. If those aren't aware, this is a very minimalistic advancer here, so hopefully I don't advance too far or something. But yes, thanks to the organizers for allowing me to speak today. I'm happy to present some of my own lab's research, but also provide a hopefully somewhat comprehensive overview of the field of how we're using induced pluripotent stem cells to be able to study and model at least certain aspects of glaucoma. And I will put that caveat out there that this is certain aspects of glaucoma, because reviewers, you know who you are. I've never gotten that comment before, though. I will start off, I guess. Okay. Just some disclosures, not even so much for disclosures because I'm not really getting anything out of these, but just to be fully transparent, there is some intellectual property that we've developed from some of the research in my lab. But the emphasis I want to provide here, for those that are not fully aware, is what exactly induced pluripotent stem cells are. Induced pluripotent stem cells, abbreviated IPS cells for short. The pluripotent term itself simply means that these cells have the potential to form any cell type of the body. So this is great because we can potentially then direct them to differentiate into any cell type that we want. So when they're derived from a donor, it doesn't matter if you're studying the retina or anything else. Don't know why you'd want to study anything outside of the eye, of course, because that's the most fascinating part, but you can, for example, study something else. These cells are generated through the use of somatic cells, typically skin fibroblasts or peripheral blood mononuclear cells, something that's easy for a patient to donate. You can imagine that would be a lot more agreeable to patients than digging around in your brain or your eye. And they're genetically reprogrammed to this pluripotent state through the use and introduction of what are called the Yamanaka factors. These are OCT4, c-MYC, KLF4, and SOX2. These are 4 factors that Shinya Yamanaka and his lab had described in 2008 to be necessary for the effective reprogramming of any kind of somatic cell into a pluripotent stem cell. Applications for these iPSCs can include things like better understanding human development, because stem cells and models developed with stem cells are intrinsically developmental models to an extent, because they're starting from this pluripotent state. We could also use them, though, to study some aspects of disease states for in vitro disease modeling to better understand, at least at the cellular level, if not perhaps now encroaching upon tissue level, what's going wrong in disease states. Also, strong potential for drug discovery. We've seen over the past several years increasing adoption of these iPSC models by pharma to be screening drug compounds and kind of bridging the gap between animal models and clinical trials, as well as for cell replacement. And I'm disappointed because Brad ran out of time to present a little bit of the data that he and I collaborate on, but it's another exciting area. So, iPSCs are great. The federal government says we should reduce our use of animal models in favor of things like iPSC models. There are a lot of advantages, but there are disadvantages or limitations as well. So, advantages, the iPSCs can provide us valuable insight into human cellular development as well as disease states, with the emphasis there on human. And I think it was Colleen before said that she's training trainees in her lab not to cure mouse glaucoma, but human glaucoma. And ultimately, what we want is to be able to build to our knowledge, leveraging information and data and results that we get from animal models, but also from cellular models like this, to provide a more comprehensive, holistic view and understanding of what's going wrong. And here we're able to look at human cells and how they're affected in disease states. So when they're derived from patients, the potential exists to use these cells also for cell replacement. So if you think of later stages of glaucoma, and I'm going to show my bias here in the more posterior side of the eye, because I'll focus on the retina with all due respect to the anterior part. But my example here, once we've lost a significant amount of retinal ganglion cells, it doesn't really matter so much about what kind of drug discovery efforts you have. The cells are lost. There will be a need for cell replacement. And this does provide opportunities then for kind of providing spare parts, as you will, to replace those cells that have been lost. However, where they fall short, and probably will for the foreseeable future, is that we cannot currently use iPS cells to mimic any kind of higher order visual processing. We're not able to look at visual processing for like transmission from photoreceptors to bipolar cells to retinal ganglion cells into the brain. Our technologies are not that advanced. To be able to do that, it is essential to have animal models to complement our in vitro studies. So this field has advanced over the years, and I was putting this together the other day, and I was telling some people in my lab that made me feel really old, because some of these studies that seem like it wasn't all that long ago are encroaching on about older than undergrads now. But one of the first studies, and remember the first example of pluripotent stem cells, before iPS cells came around, the only other example were embryonic stem cells. The first description of human embryonic stem cells occurred in 1998 by James Thompson at the University of Wisconsin. And then just within 6 years, there was a paper by Bob Lanza that came out where they were describing their ability to identify and isolate retinal pigment epithelial cells from human embryonic stem cells. And this was actually a pretty neat study for 2 reasons. One is because this picture over here, you're kind of seeing just an overgrown well of a dish with a ton of cells in there. You kind of see it looks like a big blob kind of taken over. But there are a couple of pigmented patches around there, because the RPE have a pigment themselves, kind of built-in reporter. Who needs GFP when you have pigment in the cells? You could visualize them under the microscope, and what they were able to do is microdissect out those little patches and be able to expand them in vitro and develop pretty purified populations of RPE. This is also cool, though, because there is no direction given to these cells. Spontaneously, they're able to do this also reproducibly to form RPE, so it really hinted at there was a propensity of these cells. They could form any cell type of the body, but reproducibly, there was at least some cells there that were retinal, or at least RPE. And this really hinted at what was to come for the field. A couple years later, Tom Ray's lab described the first derivation of neural retinal cells from human embryonic stem cells, led by Deepak Lamba when he was a postdoc there. In 2008, first we had a paper by Masayo Takahashi's lab from the Riken Institute looking at derivation of retinal cells and a little more advanced studies of the types of retinal cells we can get from embryonic stem cells. And that was around the time that Shinya Yamanaka and Jamie Thompson both kind of concurrently published the ability to derive human-induced pluripotent stem cells, or iPS cells. And this opened the door for studies then of getting into iPS cells with a lot of advantages there for disease modeling, patient relevance, so forth. It was also very opportune because I was a postdoc at the time in David Gam's lab at the University of Wisconsin, so we worked closely with Jamie's lab to incorporate some iPS cells into our work that led to this study here, which is one of the first studies then to show iPS cells can give rise to retinal cells. Oops, and I forgot my animations. I did have a little example of each of the cells that were differentiated through each of these methods. But all those methods were just cells grown in a dish in kind of this monolayer kind of fashion. One of the real revolutionary things was the development of what's now called the 3D retinal organoids. So these are three-dimensional structures that mimic some of the spatial and temporal organization of retinal cells forming into retinal-like tissue. So one of the first real notable ones here was this one here that was described by Yoshiki Sasai in 2012. This was working with mouse stem cells. And he, using this GFP reporter, found that they could induce the cells to adopt this retinal-like tissue, a really profound image. This was on the cover of Nature or Science, or one of those big things that we all kind of dreamed of publishing in, but so far I haven't, but one of these days. This is with mouse cells. So that was one of the shortcomings. We wanted to translate this to humans. This is also not just grown on its own. It was also attached to a mass of other cells there, so it made it a little more technically challenging, so you had to microdissect out each one of them to work with them. Some of the other studies that were kind of advancing the idea of retinal organoids was the study from David Gam's lab in 2011 showing these optic vesicle-like structures that were derived from human embryonic and induced pluripotent stem cells, here stained positive for CHEX10, marker retinal progenitor cells, and then Val Canto-Soler in her lab kind of advanced this to form retinal organoids, and here's the nice picture of some photoreceptor-like cells. Now, some of the challenges with this, though, is that this was not a pure population. There's always a lot of other stuff in there. So even in the best cases, the ones that you see around here that kind of have the more golden ring around the outside. Dave Gamm likes to say these look like Cheerios floating in a dish. These themselves are the retinal organoids, but there's always some other stuff in there. Tends to be more forebrain stuff, linking together a semi-common developmental lineage there. The neural retina formed as an evagination from the anterior parts of the neural tube. So just last year, really talented former PhD student in my lab published this paper where she looked at kind of updating some of these methods. We're forcing the reaggregation of these cells to form a very reproducible size and shape. So you see over time here, this little aggregate forming of all these cells. We can seed precise numbers of cells into the dish to get reproducible size and shape of these aggregates that eventually form very consistently sized and shaped retinal organoids. And when done properly, under the right conditions that she was able to tweak, we get this 100% of the time. Not just every batch gives us retinal organoids, but every single aggregate there forms a retinal organoid, really advancing now our ability to look at retinal-like phenotypes. So to derive the cell types in glaucoma, there's a couple of major cell types that we want to look at. Cells of the trabecular meshwork, of course, and there have been some foundational studies by Marcus Kuhn's lab and the Dew lab at Pittsburgh. I'm going to focus on the retinal ganglion cells a little bit here and some of the processes and methods that have been developed by my lab, Don Zak, Iqbal Ahmad, Mike Cheatham, and a variety of others. There's too many for me to really do diligence to all of them. There are a number of different methods that we can use to derive retinal ganglion cells. And commonly people talk about things like retinal organoids being kind of a 3D approach because they're free-floating aggregates of cells that are self-organized. And here's an example from a former student in my lab, Clarice Flagger, of these retinal organoids now with a TD tomato reporter in them to identify the retinal ganglion cells. So you see all that red signal is on the inner layers of the organoids, akin to where the retinal ganglion cells would be in the inner layers of the retina. Don Zach's lab particularly has pioneered more of the adherent methods. I put 2D in quotes here, and I'm going to say kind of strong air quotes, because it's really not 2D. They do form three-dimensional masses, but they're adherent. So that's what people mean when they say a 2D culture. As well as more recently, Carl Whalen's lab has really published some cool stuff to use transcription factors to drive the differentiation of retinal ganglion cells. There are a lot of advantages and disadvantages to each of these approaches. And really, my take-home message, because I see that timing is really right?
Yeah.
Oh my goodness, okay, I get what Brad was coming from. So to wrap up, if you want to see that chart, you can look at the YouTube recording. Which method is best? It's kind of like which model is best. We've heard a bunch of times. There's no clear answer. Really, the endpoint differences, my lab has explored this, they're really rather minimal. The retinal ganglion cells, you get out of them at the end, as long as they're put in the right conditions, that analogous conditions. Of course, if you put one in a really supportive medium and one in a really not supportive medium, you're going to see differences. Apples to apples, you put them all together, they're pretty minimal differences. The choice of your method really depends on your needs for the project. So how are we going to define a differentiated retinal ganglion cell? I think this is an important concept. We want to look for certain features. We want to make sure that we are really comparing apples to apples from our experiments as well as to others. So our choice of methodologies or how we're handling the cells is really important. Imagine if you're working with mouse models and you have some mice that are held at a 12-hour light, 12-hour dark cycle, nice and quiet, regulated temperature, everything is great. Next one has someone jackhammering next door and they're deprived of water and all this stuff. You would never consider comparing these. So we don't want to, I think it's really important to make sure that we are really calling a spade when we're making comparisons, especially across labs when we are looking for reproducibility. So some of the things we'll use to define from most simplistic assays, looking at neuronal morphologies with neroid outgrowth. We want to see that it has long, extensive neroid outgrowth. These are long-distance projection neurons. The expression of phenotypic markers, some of the markers that are commonly used here. This image from my lab, Shruti Patil, recently appeared on the annual report for BrightFocus, so I'll put that plug in there for BrightFocus. We want to see that they're highly polarized cells. So looking for some markers of axons and dendrites, we can kind of see some differences there. Axon initial segment with anchor in G, and then eventually functionally mature, either using whole cell patch clamp physiology or multi-electrode arrays.
We should probably wrap up, Jim.
Wrap up, okay.
Thank you.
Wow. So some of the uses of iPS cells would be for disease modeling, particularly using CRISPR gene editing to create isogenic pairs. So when you're looking at disease models, things like this optineurin mutation is causative for normal attention glaucoma, you're making comparisons that are solely due to that gene variant. Nothing else, no other differences between individuals. And my lab has a couple of posters looking at some other gene variants highlighted here. Looking at the compartmentalization, rather than just growing cells in the dish, we know that retinal ganglion cells are very long-distance projection neurons connected to the eye with the brain. Labs, including ours, have adopted these microfluidic platforms where we can see the cells on one side and induce their axons to extend to the other side. And this was just published about a month ago from Katya Gomes in my lab, where we could analyze axons, these long-distance axonal projections, and look at those specifically. And this is important because the primary site of injury in glaucoma is along the axon, and numerous groups, including Rob and Rick Libby and probably a bunch of other people I can't think of right now, have shown very different degenerative responses, whether you're looking at the somatodendritic or axonal compartments. And then finally, one of the things is a lot of different things. Yvonne told us this morning about how glaucoma occurs across the spectrum, and there's a lot of different phenotypes to really look at. There's a lot of things beyond IOP. One of the major things we can certainly look at now is neuroinflammatory responses. By combining retinal ganglion cells with glial cells, astrocytes, microglia, we can set up tricultures to look at inflammatory responses and how they could adversely affect retinal ganglion cells. And this will be the subject of a talk I give on Saturday. So wrapping up, we find the human iPS cell models are going to allow us to directly model glaucoma-relevant cells and human-specific disease states that we can't always necessarily capture in animal models. So-called microphysiological systems, so organoids, organs on chip, microfluidic platforms, are going to permit studies of cell-to-cell interactions better than just the cells grown in a dish by themselves. Things like RGT compartmentalization. And also the use of CRISPR gene editing to create more relevant and appropriate disease models to better understand what's going on due to glaucoma causative gene variants or glaucoma-associated gene variants too. So some of the unanswered future questions, I'll leave these here. There's a lot of area to grow. There's a lot of more questions we want to ask. And I'll leave this here, some of the selected references that if you're interested in this field, I recommend these. I also provided the PMIDs throughout, and this is really just a select list. There's a lot of really cool stuff out there, but also a lot more room to grow into this space. So I will happily answer questions afterwards.
Thank you, Jason. And our last speaker of the session is Tasneem Sharma.
Thank you, everyone. I would like to thank the organizers for having me today. So I was kind of tasked to talk about ex vivo perfusion organ cultures, specifically human perfusion organ cultures. And, okay. Oh, the green button, yes, of course. So just, I don't have any financial disclosures, but I do have a patent that I'm going to talk about from my lab. So when we talk about human ocular perfusion segment cultures or perfusion models, I just wanted to give a little bit of history, the current approaches, applications, and then going back into the limitations, the future directions, and then, of course, the literature. So actually, in vitro retinal studies have been done more than 250 years ago. Late in the mid-19th century, the first actual whole eye cultures were frog eyes, and they were done by Kuhn and Steiner. And they actually looked at frog eyes and how they could culture and look at ERG responses. And what they saw was they lost or they had degenerative responses within 1 hour of culture. So then the goal was to preserve these retinal organ or frog eye cultures for long periods of time. And they were able to do that within 24 hours with using 100% oxygenation. And then in the future, they went on to see or look at rabbit and rat studies predicting the need for proper temperature and glucose. So when we're using these culture environments, we want to make sure that we are not only using right environments, but how organ cultures can be grown in lab as well. And then came the need for vascular perfusion, because for long-term culture of these whole eye organ cultures, they needed to make sure that we could grow them over a period of time. And this was first performed by Leal and Grimes. And this has been now applied across species in rodents, rabbits, dogs, monkeys, cats, and bovine culture, as well as enabling ERG responses, optic nerve responses, as well as standing potentials as well. And this is something I wanted to actually touch on. I know Jason has given a brief overview of the iPSC culture, but over the decades, what we've been able to do is actually culture retinal model systems in different forms, which includes the dissociation of retinal explants, as well as whole explant cultures, as well as the very first innovation of the retinospheres. And then Jason also talked about right now is the formation of the embryonic stem cell cultures through the human retinal organoids, which was first identified in 2011, and over the course of the last decade or so, we've been able to grow successful retinal organ cultures in systems as well. But then when we come back to perfusion organ cultures, what are perfusion model cultures? It helps us to simulate blood or flow in the ocular tissues. It also enables us to study ocular physiology and pathology ex vivo. But mainly, it's bridging the gap between in vitro assays and clinical research. So when we're looking at understanding the barriers in vitro and taking them ex vivo, how we can rationalize drug design studies or drug delivery methods as well. But why is there a need for human perfusion model systems? We've learned a lot of benefits to actually looking at in vivo model systems in the lower species as well as non-human primates. But it is important to understand the anatomic, morphological, structural, and functional differences between various species and humans. Because at the end of the day, a lot of my colleagues have mentioned that it is human glaucoma that we are trying to treat. And so it is difficult to obtain accurate ocular pharmacokinetic studies, as well as these are just examples, and there's a breadth of knowledge or literature out there in terms of anti-drug antibodies that actually occur in human response, as well as the high cost sometimes of growing animal models long term. So there is also the need for ethical considerations and the 4 R's that Jill talked about as well. And validation beyond animal models, because 89% of drugs that go into preclinical or from preclinical drug formulations into clinical fail in neuroprotection studies as well. So we want to target degeneration in the diseased human eye, translate this research for effective drug delivery strategies, and use alternative methods so that we can decrease R&D pipelines. And so I wanted to focus a lot on human posterior segment cultures today, but I cannot go beyond saying what the first anterior human segment module cultures were as well. So the first designed anterior human segment perfusion culture was done by Doug Johnson in 1987. And then Ted Ackett and their group did whole anterior segment perfusion cultures, as well as this has been modified over the decades from Clark and al., Johnson and Barras and al., to finally, very recently, Dr. Mao's group has also shown corneal perfusion cultures. And these anterior segment culture models have been based on pump-driven systems, as well as gravity-driven systems, as well as flow reservoirs, where we can modulate all the different perfusions based on different kinds of gravity-based perfusion systems, as well as thermal flow, as well as using perfusions through flow rates as well. But however, in human posterior segment cultures or paradigms, you have limited access to patient samples. So it's really difficult to do human perfusion cultures in lab, as well as constraints on clinical procedures. And common animal models differ from the human eye based on the differences in macula, formation of proper lamina cribosa, retinal vascular changes, as well as tissue thickness and size. So it is difficult to characterize human tissue in detail. And this is very elegantly shown here, where when you look at different species and come all the way to humans, we have to understand the breadth of knowledge that we can have or pursue in vivo model systems, but also what the differences is between different species and the human system. So when we're looking at the size of the retina, the macula, the lack or the absence and the presence of macula, the retinal thickness, the mean RGC density, the cone density, and their RP layer thickness. So it's really important for us to use ex vivo human systems to not only do drug formulations, but also to do preclinical testing. And so today I just wanted to focus, and this is not a comprehensive list, but all mainly on perfusion organ cultures, not only from whole human globes, but also posterior segments to understand translaminar pressure differentials, as well as 3D printed whole eye models as well. So this was done first by Ethier and al. In 1993, and what we're looking at over here is where they use basic tubing as well as gauge pressure transducers and different pressure transducers to not only modulate the perfusion system in the whole eye, which is shown at the bottom here, as well as looking at transistance and resistance to understand how aqueous flow within the whole eye can actually result in pressure transduction systems. Next, Dake and Miller showed how you can take a whole eye globe system and put it in a bathing chamber, and then not only using syringe needle insertions, you can actually perfuse the syringe connected to the flow sensor and provide flow path for fluid inflow and outflow. This allowed them to grow this human eye globe in culture for at least 24 to 48 hours and then measure IOP measurements with an external pressure transducer. And here I'm showing you an actual simulation of how the whole eye globe looks with the inflow syringe and the outflow syringe measuring the transducers. And then you can look at compliance training and look at differences between the whole eye globe and what you would see in terms of pressure differentials as well. This is Dr. Jeremy Sivak's group, where they have taken 2 whole human eye globes and they have simulated anterior flow segments. So they have actually inserted 1 syringe into the iris and the lens, and then shown anterior flow segments to a secondary cannulation. And through that, they have looked at not only fresh control whole eyes, but also perfused eyes to show retinal architecture, and as well as the different kinds of markers for not only gliosis, but also retinal markers. This was the model developed by my lab, and where we've taken the posterior segment, so we cut the human eye globe in half and take the posterior segment, and then we actually do fluid flow rate-based simulations, and we have shown that we can actually maintain the retina under normal pressures and elevated intraocular pressure, where most of the ganglion cells are lost after 7 days of elevated intraocular pressure. Plus, we've shown or mimicked some sort of prelaminar and laminar region gliosis, as well as the clinical optic nerve head cupping as well. And then once we add the therapeutics, you can see that the top panel up here shows neuronal or axonal degeneration in normal and elevated IOP, but when we add the therapeutic, we can preserve or maintain the axonal degeneration that's occurring post-perfusion in these human eyes. Another example that I wanted to give is the PKI model, which has been now taken up by ophthalmologics, and there they actually take a human eye globe and they do simulations of different flow cell structures as well as intravitreal injections. And then they do microfluidic automation. And the way they do it is they want to look at pharmacokinetics, so they take the anterior segment of the eye and they put in a hyaluronic membrane and look at the different pore sizes and different methodology to do fluid flow. And so they monitor flow, temperature, eye movements, and the evolution of labeled protein molecules through the whole eye. So there are a lot of strengths for perfusion models. My voice is really dry today. So anyway, sorry. The human-based systems, which not only allows us to maintain structure and functional integrity of tissue, but also for looking at vascular pharmacology and acute retinal toxicity. Yeah. Wow, that was a lot better. Thank you, Diane. Okay. So precise control of pressure and flow, as well as direct imaging, histology, and physiological readouts, and also electrophysiology monitoring by ex vivo, ERGs as well. And actually implementing direct drug application without systemic influences. So you can actually look at the local ocular flow and perfusion of drug pharmaceuticals. But the limitations for these human perfusion models also looks at limited tissue viability. You need to have the human eye globe within hours to weeks. There's tissue donor variability. Obviously, the human genetics is pretty diverse. We've talked about that even in the morning. The death to enucleation delay, the cause of death, comorbidities, but that also helps us understand human glaucoma in a way, and the donor age as well. But the main limitations also include exotomy. Once the human eye is outside the body, you have exotomy, so the degeneration starts as soon as it's postmortem. You cannot really fully replicate systemic physiology, which is why in vivo models are important, the vascular perfusion, the complex setup and maintenance of all these perfusion systems, as well as the short time period of experimental window. You can have long-term in vivo studies, while with human perfusion models, it's within minutes or hours, or sometimes which we've been able to do maximal for 30 days in culture. So they're not really suitable for long-term studies, but they bring value in terms of preclinical human applications. And so studying posterior segment physiology and pathology was the summary, evaluating therapies in controlled ex vivo settings, and advancing this in terms of translational vision science as well. One other thing, I know I'm out of time, okay. 30 seconds more. But another value that human postmortem tissue brings us is actually we take corneal fibroblasts and reprogram them and make them into retinal ganglion cells after forming retinal organoids. But what that also allows us to do using human postmortem tissue is we can actually take whole human eye postmortem retinal tissue, compare it to our retinal ganglion cells that we make de novo, and we can downstream actually do spatial or comparative transcriptomic data as well to not only understand patient disease pathology, but also see what we are generating in vivo, ex vivo, or in vitro, is that comparable to the human retina? And just, just showing you, if you are interested in human tissue, organ cultures, this is just in the last 2 years, we've been able to get more than 100 pairs of human donor eyes. So it's something that we can actually do preclinically as well. And so future directions would include integration with organ chips, long-term maintenance, viability of tissue, reproducibility, and coupling with imaging and omics data approaches to not only control our environment, but efficient screening, predictive analysis, and improved preclinical training. And so applications would include model human disease conditions, investigate ocular drug delivery, study biomechanics, vascular physiology, maintain retinal function, and therapeutic strategies. Take-home messages: be mindful of donor variability, limited tissue variability, and oversimplification of in vivo physiology. And at the end of the day, that's what everybody's been saying, the best model depends on your research goal, ultimately. So no single model is perfect. Use complementary approaches to maximize translational insight. And thank you.
Thank you. We can now open it up to questions for the panel. So do we have our mic runners for this session? We have a question up here on the right. We have one on the left. I'm not sure who else is supposed to be.
I'm bringing the microphone.
Maybe bring the mic to this person over here too. Okay. Okay, yes.
So I actually have two questions. One for Tasneem. If I'm pronouncing your name wrong, I'm really sorry. And then one for her, I wanted to ask, when we are getting human perfused eyes for working for this model, won't we have a huge variability in age? Because sometimes we get a human donor that's maybe young and a human donor that might be older. And then would we also not get different stages in glaucoma? Because even some human donors that have glaucoma, they might pass away much quicker, and then you get the eye. So won't there be a huge variability with that?
Yes, thank you for that question. That's a very important question. Yes, we do actually have a huge amount of donor variability. We have to do almost 10 pairs of human eyes to even see any sort of subtle differences sometimes. And we do get them. Our youngest donor in the lab that we got is 18-year-old, and then the oldest one is, I think, 99 years old. But we do have, we reprogram all the corneal fibroblasts that come. So right now we have a repository of 100 donor eyes that we and corneal fibroblasts that we've reprogrammed. And so we do sometimes see age differences, and it is age-related neurodegeneration as well. So it depends on the decade. So from each decade of life, from 40 years onwards, we have almost 4 to 5 pairs. And so sometimes we do, but they're not distinctive.
Okay, thank you so much. And I have a question for Jason Meyer. I probably think you might talk about this in your long talk on Saturday. But when you have organoid, how do you shift it to be a glaucoma organoid? How do you differentiate between those cells? I understand how you differentiate between an RGC and an astrocyte and a microglia, but how do you differentiate those between disease and non-disease?
Yeah, so that's a much easier question to ask than it is to answer. As Yvonne said this morning, there is just a spectrum of different things that can contribute to the disease we call glaucoma, if we can even say it's a disease. So there are a variety of things you can do. The simplest thing, the more traditional way to look at anything related to glaucoma in an iPS cell model is to look at gene variants that are associated with that. And through the use of deriving these iPS cells from patient donors, You can look at how this affected, particularly when you use CRISPR gene editing to create matched isogenic pairs. You can then look at some of the differences there, ideally from causative mutations. So I did very, very briefly mention this optineurin gene mutation, which is causative for normal attention glaucoma. We know it leads to very severe degenerative phenotype in RGCs, which is why my lab is focused on that. But there's also a strong opportunity then, based upon all the glaucoma genetics that are out there, to really explore how this works. But then there's other things, so neuroinflammatory components. You look at that. You can look at axonal transport defects, perhaps, look at that, and kind of piece the things together. But yeah, there's not one comprehensive model in an IPSO, I would say.
All right, is there a question back here? Yes.
Oh, great talks. I enjoyed a lot. I have a question for the Gillian. I'm very curious about the heterogeneity in phenotypes. So as you pointed out, the genetic models like your LTBP2 model mimics the certain aspect of the human glaucoma pathology. So if we find the neuroprotective effect using those mice, it is fantastic from the perspective of translational research. But at the same time, those kind of the variability or heterogeneity might be a major barrier to study the neuroprotective effect efficiently or effectively. So are there any ways to adjust those kind of the variability or to consider this kind of heterogeneity?
Yeah, that's a great question. And so I think one of the things that I looked at and I mentioned is that that's actually an opportunity, right? So it's like, why are these animals different from one another with this single mutation? So that as omics resources improve, we're able to start to tease some of that apart. But from a very simple perspective, when we're looking at responses to a treatment intervention, for example, what we've been disappointed with in the past is when we do a sort of back of the envelope, until you do the study, you don't really know what your power calculation is, but we kind of guesstimate it from a few animals. And then when you do a lot of animals, it doesn't always play out according to what your expectations are. But that's especially when you're looking narrowly at things like pairwise comparisons or between group comparisons. I think ultimately these are such complex processes in glaucoma that you're really needing to model that statistically in a complex way as well. And ultimately, no matter how we slice and dice it with our animal models that we use, whether they're mice, whether they're cats, whether they're dogs, IOP, sorry to the people at the back of the eye, but IOP ultimately drives everything. So you can't look at these neuroprotective effects in isolation from the IOP. And no two humans are going to have the same IOP profile. So that is where the translational relevance comes in. And so a simple pairwise comparison of a single parameter just doesn't capture it.
We have probably time for one more. Ravati?
Yeah. IOP does drive everything. So Jason, a question for you on that. Are there good retinal ganglion cell iPSC models where they can actually model increases in intraocular pressure, or can you artificially pressure these cells? Are there good models out there to do this?
There have been. So that's something harder. So when you have cells in a dish traditionally, to really mimic that kind of elevated pressure in a physiologically relevant way, rather than just kind of evenly across everything, has been challenging. That's why the field has kind of started off with things like glaucoma gene variants and so forth. As the technology develops and we're able to develop better models like the microfluidic platforms to isolate some of the axons. The field of biomechanics advances also. Some of this, we do see some of this starting to merge together, at least conceptually. I've not seen it come to practice yet, but I would imagine over the next several years, we'll start seeing more challenging aspects of glaucoma being integrated into these IPSL models.
Great. Oh, one more comment.
Yeah, I think it's useful for the sort of new peeps to the field to consider two really important caveats. One is that using hydrostatic pressure in culture is actually oxygen glucose deprivation. It's not pressure. There's a great paper by Ross Ethier and colleagues that you should look at in IVS. And then the other one is about the human eye, the bias of getting human eyes. So you're going to tend to not get early glaucoma because you look at an eye, typically there's not a lot of good records, even if you consented to get the records. And so you either look at the eye and say, yeah, that's a glaucomatous optic nerve, or you get some weak records that say, yeah, the person was taking timolol for 20 years or something. And so you tend to exclude eyes that are in the middle zone that's otherwise probably very interesting. You get hypernormal eyes for your normal group, and you get sort of more advanced glaucoma for your glaucoma group. It's an important caveat to consider.
All right, well, let's thank our speakers one more time.